Assalamu alaikum friends, welcome to my channel MBBS and BDS lectures. Today our topic is related to histology, steps of histoslide preparation. Before going to explanation, if you want to watch the same topic in Urdu Hindi, I already upload the same lecture on my YouTube channel in Urdu Hindi. Go and watch that video. But if you want to watch in English language, then this video is for you. Now, steps of histoslide preparation. There are 4 steps of histoslide preparation. Number 1 is fixation. Number 2 is tissue processing. And number 3 is sectioning. And number 4 is staining. First we start from fixation. First we collect a tissue specimen from human or animal. Then we should immediately transfer this collected tissue specimen in fixation stage to 10% neutral buffered formalin solution for 24 to 48 hours. Why in fixation we immediately transfer collected tissue specimen to 10% neutral buffered formalin solution for 24 to 48 hours to prevent autolysis of specimen. To prevent autolysis of specimen mean to preserve the specimen. For preservation in fixation stage, we transfer the collected tissue specimen to formalin solution for 24 to 48 hours. This is the first step of histoslide preparation. Now come to second step. The second step is tissue processing. Tissue processing means now the tissue specimen pass through a long process. In tissue processing is done using a paraffin block in 3 steps. Now we do tissue processing by using a paraffin wax block in 3 steps. Mean there are 3 steps inside tissue processing, or tissue processing is consist of further 3 steps. Number 1 is dehydration. In dehydration we transfer the specimen to high alcohol concentration in order to remove water from specimen and formalin from specimen, which are used in first step. In dehydration, specimen immersing in high alcohol concentration to remove water and formalin from tissue. This is called dehydration. After dehydration in tissue processing, the second step is clearing. Clearing means we remove, like in first step dehydration, we remove formalin from tissue by using high concentration alcohol. Now in clearing, we remove the high concentration alcohol by using xylene. To remove formalin we use alcohol. To remove alcohol we use xylene. In clearing, an organic solvent like xylene is used to remove alcohol, and the second advantage of xylene is and help in infiltration with paraffin wax. Infiltration with paraffin wax is the second advantage of xylene. The first Action of xylene is to remove high concentration alcohol, and the second advantage is help in infiltration with paraffin wax. This is called clearing. The third step in tissue processing is embedding. In embedding, the tissue specimen is now in embedding we embed the specimen into molten paraffin wax container at 60 degree centigrade. In embedding, we embed the tissue specimen in molten paraffin wax at 60 degrees centigrade. This black line is show a container, and the blue dot show a molten paraffin wax, and the red is a tissue specimen. In embedding, we place tissue specimen inside a container in which the molten paraffin wax is present at 60 degree centigrade. After this, we place this container into refrigerator where a cubical shaped paraffin block is formed due to pressing up molten paraffin wax, and inside this block there will be a tissue specimen. This is called embedding. Now tissue processing is complete, in which we studied the first step dehydration, second is clearing, the third is embedding. Now the third step, sectioning. In sectioning, we make a thin section of cubical paraffin block. In sectioning, first we remove the surface paraffin wax from paraffin block to expose the tissue. Like this is a paraffin box, cubical block, and upper to this tissue the paraffin wax is removed in sectioning. Remove surface paraffin wax by a machine called microtome. Then in sectioning, when a tissue is exposed on the block, on paraffin block, then we cut a thin section of paraffin wax with tissue by microtome, like this. This is a thin section of paraffin wax with tissue which thickness is 3 to 10 micrometer thick. Now this thick section 3 to 10 micrometer with tissue will transfer to the warm water bath. Why? Because we know the wax is dissolved in warm water. Like in prosthodontics in dentistry, we dissolve the wax in warm water. Now we transfer this 3 to 10 micrometer thick section of paraffin wax with tissue to the warm water container. This is the black show container, and the inside container the black dot is a warm water, and the blue is a paraffin wax which dissolve inside the water, and the red is a tissue specimen. Now transfer thin section to warm water bath where wax dissolve and tissue floating on surface, and tissue will be floating on surface in warm water. Then after this we remove the tissue from warm water and to dry. When it dry, it help in adhesiveness to slide. It will be easy adhere to slide. Now tissue transfer to slide. This is a black slide and the red is tissue. This is the sectioning process. After sectioning, the last process is staining. In staining, the first question is why we stain the cell, why we stain the tissue, why we stain the tissue specimen. Because cell structure are colorless. We cannot differentiate without staining. Now in staining we use H and E stain, hematoxylin and eosin stain. We place this tissue in a container. We place this tissue in a container of eosin and hematoxylin stain, where the nucleus got blue color and the cytoplasm and cell membrane got a pink eosin color. This is a staining. After that in staining, we place this tissue in a cover slip. This is cover slip. This is cover slip, which show in a black. On cover slip, we use a sticky material and then place this tissue specimen, the tissue in a cover slip, and the sticky material is applied on cover slip, and then the tissue will be packed in cover slip, and now this tissue is ready for examination. This is the whole process of step of Histoslide preparation. Now, if you like my videos, press the like button and share these videos with your friends. If you new to my channel, subscribe my channel and press the bell icon in order to you receive every new video notification. For watching my videos, I am very thankful to you. Thank you very much.